Transcriptomic is a very powerful tool in research. It gives us the profile of RNAs that exist in a sample. Genomics gives us the genes that exist in a sample, and transcriptomic tells us which ones of them are active. My name is Pedro Faísca, and I'm the head of the Histopathology facility here at the IGC. The added value to have this technique here at the IGC is that we have multidisciplinary facilities that are working together, and so we can do all the process on the same place. So we can process the samples here on histology, do the sectioning, then the genomic will evaluate and do the transcriptomic. As a pathologist, I can help them understand by saying this is a tumor, this is stroma, this is the inflammatory foci, and help them to understand what's really happening. So it's the full process in the same place. Single cell, it tells us on a single cell the genes that are being active, and the spatial transcriptomics gives the context, the spatial distribution of those genes, so we can tell where those genes are being active.
This can be done, and originally it was done in samples, frozen samples, but the biggest breakthrough came recently where we are able to perform it in paraffin preserved samples. In all of the hospitals, and mainly in our work in histopathology at IGC, most of the samples that we process are in paraffin. So this allows us to have a large biobank of resources of samples where we can go back and look at them through a different eye using this technology, which is very, very important. The ability that this technology allows us to go back to samples that are preserved in our biobanks is, for instance, we can go to samples from tumors in patients and we will be able to see what the tumors were expressing, and this might indicate how or why they responded differently to chemotherapy, radiotherapy, and how the outcome was different even though they had the same tumor in the same stage.
So imagine a researcher studying cancer. So in a cancer with this technique, we can know which genes are active in the tumor cells, but also in the nearby stroma, in the inflammatory foci that's surrounding the tissue. So with this spatial information, we can understand in more detail what is really happening in that process. So in cancer, this is one of the techniques, even for clinical translation studies, this is where it's going to be huge in the future.